Hello, MRS participants, and thank you for attending my virtual talk. On this talk, I hope to present something a little bit different than the other things that were presented in this session, as most of the antibacterial particles that are presented are synthetic and need some complex fabrication. However, in this talk, I'm going to present you with a work done in our laboratory where we utilized natural clay particles for the selective delivery of antibacterials to selective target bacteria. The motivation for this work and the use of nanoparticles for antibacterial activity is probably quite understood by now. However, in this work, we emphasize the need for a selective targeted delivery of antibacterial, so that in the treatment, we will eradicate only the pathogenic bacteria and would not harm the entire flora of our body, leading to the unwanted condition of dysbiosis. The concept is to use natural clay that have tubular morphology, equip them with antibodies onto their surface, and load them with antibiotics. So these antibiotics will be released only at the proximity of target bacteria in a suspension. First, I would like to introduce you to the fascinating world of natural nanoclays. These natural materials are minerals that are just mined from the earth and sold by the ton. We have kilograms of them in our lab. They are composed of alternating layers of silica and alumina, and specifically the nanoclay we are using is called Halloysite nanotubes. Hopefully you're all Googling it as I talk. These clay have a unique tubular morphology, which is completely intrinsic and natural, which is ideal for the loading of various compounds. In terms of biocompatibility, it is very tempting to identify HNTs, Halloysite nanotubes, as biocompatible. However, the truth is that not enough research has been done till now. In a thorough review that we have recently published, we do some comparison between HNTs and other common high-ratio nanomaterials, such as multi-wall carbon nanotubes. And we have found that at least HNTs are not more toxic than the carbon nanotubes. Together with the other advantageous properties of HNTs, there has been a surge interest in utilizing them for many applications, such as fire retardant and in polymer industry, in ceramics, but also for biomedical applications, which are much more exciting for us. These biomedical applications are based on the very natural interactions between HNTs and biological cells, starting from the very basic colloidal forces and developing into more complex interactions with the cells. In our work, we focus on the interactions between HNTs and bacteria, and we like to guide them against specific bacteria. The question is, are HNTs intrinsically antibacterial? And here also the answer is under debate, as we found in our review that there are all kinds of evidence in the literature. Some of them are even contradicting. And the conclusion is very dependent on the concentration used and the method used to assess the antibacterial activity. Sorry. In our experience, according to years of investigating HNTs, we found that at least at the concentration we use, there is not enough or substantial antibacterial effects of HNTs, and thus some modification is needed. The design we propose to use HNTs for selective delivery of antibiotics is to equip their surface with antibodies and load them with the potent antibiotic ciprofloxacin, then examining or investigating the binding capabilities of these modified HNTs. Do they actually bind to the target bacteria, which is in our case E. coli, a model bacteria? And then combining the release of the antibiotics and the binding capabilities, my aim in this talk is to convince you that indeed a selective antibacterial activity was achieved against a selective and specific target bacteria in a heterogeneous culture. So let's start with the surface modification with our raw material, which are the natural HNTs. On this slide on the left, you would find the relevant FTIR spectra for each synthetic route. The first step is the etching, acidic etching of HNTs, eliminating the alumina compound from this natural clay and leaving only amorphous silica, exposing more hydroxyl groups and increasing the surface area by 5-fold. The exposed hydroxyl groups are then aminosilicylated for higher chemical activity, carboxylized for higher hydrophilicity, and then using a rapid sulfur NHS reaction, we conjugate protein A onto the surface of the HNTs, and this protein acts as an anchor for antibodies to be immobilized at a correct orientation onto the surface of HNTs. The next step is the loading of these antibody-modified HNTs. This is simply done by mixing them together in a concentrated solution of ciprofloxacin. And indeed, a sustained release profile was measured when measuring the released ciprofloxacin from these modified HNTs. Going back to the antibody modification, we ask the question, is it effective? For that, we simply mixed the HNTs with target bacteria and non-target bacteria in a heterogeneous culture. Here under the microscope, we could see the red fluorescent target bacteria, the E. coli, and green fluorescent. For this sample, the HNTs are not modified with antibodies, and so you could see that all the compounds are just separately dispersed in the mixture. However, for HNTs that are modified with antibody, the target bacteria are specifically and selectively co-aggregated with the particles, whereas the non-target bacteria, the green ones, are just all over the place without selective binding. Moving forward to a more challenging heterogeneous culture with 3 bacteria, Pseudomonas fluorescens, which is gram-positive, and Staphylococcus epidermidis. And here the E. coli, the target bacteria, are labeled with green. And the same phenomena is observed for the selective coaggregation. Under higher magnification using STEM, we could clearly see the intact E. coli cells attached to the bundles or aggregates of HNTs, and here you could clearly see the opening mouth of the tube. For a more quantitative analysis, we utilized imaging flow cytometry, yielding tens of thousands of images of flowing objects, suspended objects, and now we need to differentiate between bound bacteria and free bacteria. This was done by image analysis. We could differentiate between bound bacteria and free bacteria. And these were counted for each bacteria type because they were all labeled with different colors. And the results are summarized in this plot. Here, for antibody-modified HNTs, whether loaded with the ciprofloxacin or without the loading, it doesn't really matter the result. We could see that the occurrence of bound E. coli, the target bacteria, is 5 times higher than the non-target bacteria Pseudomonas fluorescens and 2 times higher than the non-target bacteria Staphylococcus epidermidis. And thus we conclude that indeed the antibody modification of the surface of the HNTs endowed them with selective binding to the target bacteria. And now for the most interesting part, whether this binding actually translates to a selective antibacterial activity. This was investigated in a rather simple assay. The heterogeneous culture was just spread on plates on 3 selected media, 1 for each bacteria. And I remind you that this is a heterogeneous culture. All the bacteria are together. And indeed, in the presence of the particles, we could clearly see an inhibition of the growth of the target bacteria, the E. coli, whereas the non-target bacteria, the Serratia mersicens and Pseudomonas fluorescens, were not inhibited to that extent. However, it is very important to note that each bacteria has its own intrinsic susceptibility to C4-fluoxetine, and therefore the results has to be normalized. And here are the normalized results expressed as an equivalent C4-fluoxetine concentration. In this plot, you could see that using the correct particle concentrations, we clearly see that the inhibitory effect exerted on the target bacteria, E. coli, is 2 times higher than the non-target bacteria, Serratia mersicens, and 10 times higher than Pseudomonas fluorescens. Although all the bacteria actually experienced the particles together because they were all in the same co-culture. This leading us to the conclusion that indeed a selective activity was realized by modifying the HNTs with antibodies and loading them with antibiotics. The proposed mechanism is starting with the heterogeneous culture, the selective binding of the HNTs to target bacteria, and then the suspended release of the antibiotics probably occur mostly at the proximity of the bacteria, the target bacteria, leading to the selective antibacterial effect. In the future, this kind of preparation may be used to treat gastroenteritis, where the protection of the microflora is very important. We actually need to target only selective bacteria in this challenging culture, core culture. And with this optimistic tone, I would like to summarize this talk and reminding you that natural Halloysite nanotubes are natural nanoclays mined from the earth that could be used for carriers of pharmaceuticals. In this example, we modified them with antibodies and realized selective binding to target bacteria. Later on, we loaded them with antibiotics and showed that indeed a selective antibacterial effect was achieved. In the future, this kind of method or system could be used for treating gastroenteritis and also combined with trigger mechanism or even directed to any other microorganism with just changing the antibody or the targeting probe. Thank you very much for your listening and attention, and I would like to thank my group for the support and the Azrieli Foundation for a great support. And with that, I conclude this talk, and thank you for your attention. You are welcome to ask questions further on. Thank you very much.